Now today's class would be on separation and isolation of the DNA fragments. Now since we were dealing with recombinant DNA, recombinant DNA means the required DNA that is artificially engineered. Okay. Now this separation and isolation of the DNA fragment could be done through electrophoresis. Now what is electrophoresis? Basically it's a technique, it is a technique that used to separate the DNA fragment. You see, it is used to separate the DNA fragment after they are cut. Now how it is cut? It is cut through the restriction endonuclease. Now once it is cut, this simply make use of an electric field. You see, electrophoresis, that means they are separation of the DNA fragment using an electric field and a medium. They need a medium. As we do in electrolysis, we need an electrolyte, and here instead of electrolyte we use a medium or the matrix, which is generally, what is the medium? It is known as agarose. What is agarose? Agarose is a gelatinous extract. It is a gelatinous extract from an algae or seaweed. As algine, it is also there. Agar is there. These are all gelatinous extracts from the algae, and hence the name comes as gel electrophoresis. Gel means a medium, agar medium or a gelatinous medium, and when electricity is passed through this gelatinous medium, the DNA fragments separate. Now DNA is basically, it takes up a negative charge. DNA is negative charge, so obviously it moves to the anode, that is positively charged, and then these DNA fragments, they are sieved out. What is sieved out? These fragments passes through them, and through this process, the agar medium, the smaller fragments, you see, the smaller fragments move further away, while further away from the anode. Okay, further away from the anode, while the larger fragments, since their mass is since they are heavier than the smaller one, they are not much deflected and they are found near the anode itself. Now these DNA fragments are not visible. You cannot see these DNA fragments like that because they are very small in the naked eye. So they are stained. They are stained. See, in lab you see that the staining material are used to stain the nucleus, to stain the cytoplasm. So the stain here is used, that is known as ethidium bromide. Ethidium bromide, and it has to be visible under the ultraviolet light, not the normal light. Okay, and when seen under this ultraviolet light, you can see orange bands. Okay, orange bands of DNA are clearly visible. Now if you see here you see this is a DNA, this thing, see, smallest fragments are deflected, and these orange bands, a typical agarose gel electrophoresis, showing the migration of undigested, that means they are not digested. Since they are not digested, they could be separated easily. Okay, so this is basically the method that is used. Okay. Now once this DNA is separated, next is we need a cloning vector. Now what is the function of the cloning vector? This cloning vector uses either a plasmid or a bacteriophages, and that has the ability to replicate. They have the replicate within the bacterial cell, and it is independent of the control of the chromosomal. See, chromosomal DNA is within the nucleus, and extra chromosomal nucleus are known as plasmids. So plasmids have the capacity to replicate, and it is not under the control of chromosomal DNA. So they can make copies of 50 to 100 per cells. So if the alien DNA or this DNA fragment that is cut off from the required quality, that single DNA, if it is linked with the bacteriophages or the plasmid, then we can multiply with the equal number of copies of the plasmid and bacteriophages. So for that vector is used. And what is vector? Vector is, they are engineered. in such a way that they help the easy linking of the foreign DNA and the selection of the recombinant from the non-recombinant one. See, what is not required forms the non-recombinant variety, and what is required is known as the recombinant variety. So recombinant and non-recombinant has to be identified and has to be separated from each other. So this non-recombinant variety is denatured. It's eliminated from the system and the recombinant goes into the required DNA. Now here you see what is the features. What are the features that is required to facilitate cloning? What do we require? First, we need the origin of replication. What is origin of replication? That is known as ORI. Then we need a selectable marker. What is selectable marker? That is known as a transformant. What is this transformants? Transformers or transformants, what are these? Because these help these replicated DNA to get adjusted to the cloning vector. Okay, and cloning site. Site means it is the position where this DNA had to be inserted. So that is also a very important part. Now, the origin of replication. What is origin of replication? Origin of replication, ORI, is basically, it is a sequence. Okay, it is a sequence. Mind it is nothing but a sequence from where? From where the replication starts, and any piece of DNA when linked to this sequence can replicate. Did you get it? See, sequence from where the replication starts and any piece of DNA, that means this recombinant has to get adjusted to this piece of DNA, and so when it is linked, this sequence can replicate. Okay, now what are markers? Markers are used as identifier. See, in the board, if I am writing 1, 2, 3 with this colored pen, so this colored pen tags that it is the important points. So like that, you know, we don't use colored pens, but we use certain enzymes. Okay, so these enzymes are tagged and they are required to identify and eliminate non-transformants. Non-transformants means that is not required in the and the selective by permitting transformation. That means what is not required is non-transformed, and what is required, they are selectively permitting only that portion that is required to transform. And what is transformation? Transformation is also a procedure. You see, transformation is not a DNA sequence. It is a procedure that introduces the DNA fragment to the host bacteria. So it is a procedure, it is a method. Okay, now when we are using that, we need a very important thing, that is a cloning site. This is very important. What is cloning site? Cloning site is the particular position, okay, where the vector needs to identify the site of a single recognition site. That means the vector that or the transformation, the procedure, they need to identify the position where the DNA has to be inserted, and that is done through restriction enzymes. Okay, so the 3 basic points are here. Now see, if you can see the cloning site here it is clearly written that in order to link the alien DNA, the vector needs a very few, preferably single recognition site. Now what is single recognition site? That means the place where the DNA has to be inserted. Okay, now you see the presence of more than one. What would happen if more than one recognition sites are there within the vector? It would generate several fragments, which is not required, and it would complicate the gene cloning. Okay, so the ligation of alien gene, what is ligation? Fixing or addition. DNA is carried out by the restriction site present in one of the 2 antibiotic resistant gene. Now what is antibiotic resistant gene? We will come to it later. Now for example, you can ligate a foreign DNA. Okay, you can ligate a foreign DNA here. You see HIND3, this is a site. Okay, EcoRI is an enzyme. Okay, and here that is BAM-HI. See, you will find here they are site of tetracycline resistant. See, this portion is tetracycline resistant. That means what? If the this part of the fragment DNA, if it if it is kept in tetracycline, it will grow. Okay, but if it is kept in an ampicillin antibiotics, it will not grow because it is tetracycline resistant. That means it will tetracycline will not affect. Now see, antibiotic resistance, for example, BAM H1. H1 is for Hind1, we had learnt it earlier. Now it is tetracycline resistance gene vector. This is the chemical name of the vector. The recombinant plasmid tetracycline resistance due to insertion of the foreign DNA, but they can select from the non-recombinant one by plating the transformant in tetracycline containing medium, ampicillin containing medium. So you see here, this is the ampicillin resistant medium and this is the tetracycline resistant medium. Okay. Now you see, on plating this, now the recombinant. See, the recombinant will grow in ampicillin medium. Now if tetracycline ampicillin resistant, it will grow in but not in any other medium containing tetracycline. So in this case, one antibiotic resistant gene helps selection of the transformant, and whereas the other antibiotic selects the other. So you see, all the genes get selected and ultimately this vector is formed. Okay, and what is that? Here, this is ROP. What is ROP? ROP actually codes for the replication of the plasmid. Okay, so if this is coded and this ROP gets completed, so that means it would be coded. And this again, we will be dealing this portion, we will do it in our next class in further detail. Okay.